Thanks. It's a great honor to be part of this session. My name is Will Parsons. I'm a pediatric oncologist at Texas Children's Hospital and Baylor College of Medicine. In my spare time, I'm also a cancer genetics and genomics researcher. And I'm going to be talking today about a clinical study that we're doing at our institution to try and both set up the infrastructure to do clinical genomics and also hopefully allow us to start to answer some of these questions that have been so excellently pointed out already. So the overall goal of our research program is obvious and is similar to others who are trying to use genomics in these contexts. And that is, we're actually trying to use them to provide real-time genetic information relevant to the care of both the child with cancer as well as potentially other family members. And one can come up with an infinite list currently of challenges to that, practical limitations, starting with the fact that the vast majority of variants that we identify through tumor sequencing, we have very little ability to interpret, particularly what their relevance might be for the clinical care of a patient, let alone just the biology of the tumor. The second, we have a limited number of preclinical models such that when we have variants we're actually interested in and on paper make perfect sense, we have a limited ability to study those. Because of those first two, a relatively small number of drugs, although the number of molecularly targeted agents is exponentially increasing. Fourth, and that's actually what we're going to be focusing on here a little bit, is we don't have any experience with trying to apply these technologies. So both how to explain them to patients, how to explain them to oncologists, how to interpret the results, how to understand the kind of the spectrum and compendium of potential clinically relevant findings we see. And then fifth, it's actually a challenge for all of oncology in terms of incorporating genomics, but it's particularly relevant for pediatric oncology where we have smaller number of patients. Once you get down to the point where you're subdividing specific cancer types into multiple different molecular subtypes, trying to do clinical trials to prove that specific therapies work for specific rare tumors, for specific rare subtypes of tumors gets to be a real challenge. So despite all those challenges, we decided to go ahead. So this is what we call the Basic 3 study. It's a study on clinical tumor and exome sequencing. So Baylor College of Medicine has a clinical genetics lab that does germline exome sequencing in the clinical lab. They also do tumor exome sequencing. And the idea was to try and start to identify the challenges and begin the process of incorporating this into the decision-making for our patients. And then secondly, to start to interpret and evaluate the actual clinical utility of this data and the utility of the data for oncologists as they take care of their patients, and most importantly, how the families feel about its utility and their preferences for receiving types of genomic information. Sharon Plon is a cancer geneticist at our site who's the co-PI of the study. And this outline basically shows how it goes. So over a couple years, we're enrolling roughly 280 to 300 children with newly diagnosed solid tumors. So they can be brain tumors, they can be non-CNS solid tumors, basically any type of cancer. It's not for a specific histology. Blood and tumor samples are obtained from the patients. They go to the clinical sequencing lab. The lab, just as they would for a patient who was ordering the test from outside of our study, they get back a germline and somatic mutation reports. Those results actually go into the medical record, electronic medical record at our hospital. The results are given back to the oncologists who take care of that patient and explained as need be. And then in conjunction with our study genetic counselors, we have 2 genetic counselors who virtually work full-time on the study, the results are then given back to the families. And then it's not an interventional study, so we're not dictating what treatments the patients should get. The idea here is we're providing this sort of information and then seeing what the oncologists actually do with it. And then we follow the patients for several years after diagnosis to see both whether the oncologists actually change treatments for those patients. And in pediatric oncology, most of the upfront treatments are relatively defined. Most of our patients go on clinical trials. But the challenge really comes when their tumors come back and at that point what treatments might be used. So this is basically the study. The inclusion criteria are very broad. We're essentially taking all comers. If they have their surgery at our hospital and we're going to be taking care of them there and they have a solid tumor, we enroll them. We also enroll the parents of the children because we both ask for blood samples from them for clinical purposes to help interpret germline findings that we see. But then also on the research side, where I'll allude to in a minute as we're doing other analyses, it's helpful to have the parental samples as well. But then we also have a series of surveys and interviews of these families at various time points before they get these results, after they get them back, several years down the road, to try and see from their perspective whether we're actually helping in the care of their child and family. And then we also enrolled all the oncologists for the children. So there's 16, roughly, pediatric oncologists who take care of these patients at our hospital. So there are many clinical and research questions that we're trying to answer here. The first are related to simply the clinical questions of what do we see in these types of data? How often can we find even theoretically clinically relevant findings? And so on the tumor side, obviously we give these tumor results back to the oncologists and families. And then in a variety of ways, including observing what decisions get made for the patients, as well as surveying the oncologists and also assessing for ourselves what we think the utility of the data should be, we try and get some idea of what we're finding in those tumor exomes. Similarly, on the germline side, we're measuring the frequency of both incidental or non-phenotype or non-cancer related findings, but then also how often we find pathogenic cancer susceptibility mutations that have some implications for surveillance testing, for screening of other family members. Because as we're doing genomics and doing untargeted sort of testing, these are all the types of data that we can find. There are a number of goals that I'm not going to touch on today related more to the sequencing itself, the methods being used for that, as well as the, for example, reporting platform. So there's an iPad app developed that could be used to explain these results to the oncologists as well as to family members that are somewhat interactive and hopefully do a little bit better than just a printed paper copy of a report. But then also developing better methods to annotate these data. And then finally, we have a group of ethicists and social scientists who study doctor-patient communication who are actually trying to assess, as I said, the preferences and opinions and ideas of both the oncologists and the families, as those are the people actually involved directly in the process for this type of data. So for example, the visits where the genetic counselor and the oncologist give the results back to the families are audio recorded. And some scientists who study doctor-patient communication and interpretation and explanation of genomic data code those and then are trying to do various analyses to see what types of patient questions the families and patients actually ask about this data, and equally importantly, which parts of them, because it's a complex set of data, which parts of them do the oncologists actually think are important in terms of how they explain them to patients. So there's a number of those types of goals as well. So thus far, we've enrolled, actually, this shows 150, another 10 in the last couple weeks, 160 patients. Of the families we've approached, roughly between 80 and 90% are very excited about or agree to participate in a study like this. Those who don't, most often it's just because of the circumstances of the diagnosis and the initial decisions about treatment for their child, and really just not willing to be involved in one other thing, which is perfectly understandable. There are actually very few of the families who object or don't want to participate in a study like this because they don't want to know about genetic data, or because they have concerns about the privacy risks of having genome or exome data in their child's medical record and the implications that might have for insurability, privacy, et cetera. And then there's very few that just don't want any additional blood drawn on their child. But in general, this points out one challenge we have is that the enthusiasm from parents and patients for this type of precision medicine approaches is huge. And they don't understand all the challenges that we face and all the limitations that they currently are. So when we're talking to one of our families, one of our first jobs is actually to explain, look, these are the exciting kind of technologies and data that we're going to try and generate on your child's tumor and blood. And we hope in the future to learn how these might help take care of your child. The fact of the matter is that now, at this current time, there's such a number of limitations that for most it's not going to be of any use yet. So this shows just kind of the broad spectrum of diagnoses that we see. This was the diagnosis for the first 100 subjects enrolled on the study. About a third of the patients have central nervous system tumors of various histologies. About 2/3 have non-central nervous system tumors. And we have tumor available for sequencing in the majority of these. So almost 90% of those with the solid tumors, we have tumor, meaning we can do tumor exome sequencing in addition to blood exome sequencing. For the brain tumors, it's more like 70%. And patients are eligible for the study if they don't have any tumor available, because then we can just do the blood analyses. So first, I'm going to talk a little bit about what we're seeing from these tumor exome results. I'll give very briefly some of the methods, although I won't go into any great detail. It's being done, the sequencing done in the clinical lab on an Illumina pipeline, on paired tumor and blood samples in one HiSeq lane. This just shows some of the data and coverage data for a recent set of patients, roughly, almost 19 gig per patient sample, a mean coverage of between 250 and 300x for the germ and the tumor line, germ line and the tumor respectively. These are data are generated in terms of variant call format and lists of mutations and annotation as they were essentially in the research sequencing lab. So in terms of mapping to the exome, doing various annotations and call variants and QC filters. The tumor and normal VCFs that are generated then go into a somatic mutation caller and get further annotation to identify the list of somatic or tumor-specific mutations that are present. And then also, there's a defined set of cancer genes that analyses of the normal germline are also performed. The steps after that come into the molecular pathology lab, where, and after generating these data, applying the filters, getting mutation lists, then the job of the pathologist is to interpret and rank those, do additional literature searches, et cetera, confirm them, initially done with Sanger sequencing, going to be done now on using actually Ion Torrent, and then developing the reports for the families. So the first challenge for the pathologist is trying to give some indication to the oncologist of well, what are the important results here? So in a somatic mutation report, for example, it's as for other mutation or other types of clinical reports, so an MRI scan, a cytogenetic study, et cetera. In some ways, it's essentially just an annotated and explained list of data and variants. So this is very roughly speaking how the mutations, the somatic mutations in the report are broken down from categories 1 through 4, in theory, in terms of decreasing potential clinical utility. So category 1 mutations are those where if you found this mutation, there is known to be clinical utility for that mutation for the specific tumor type tested, meaning essentially standard of care is already to do this specific gene testing, and it helps either determine prognosis or determine what therapies might be used. The fact of the matter is these are incredibly few and far between currently. The vast majority of even potentially clinically irrelevant things are in a less clear category that the lab categorizes here as category 2. And these are cases where, similar to some that you heard from the previous talk, based upon what's known about the gene, what's known about that tumor type, they are potentially linked to either FDA approved or drugs that are in clinical trials. So specific gene pathways, functional groups that are known to be in theory targetable in some way. And this is actually the area where the oncologists are most interested in the data at the moment. The category 1 mutations are already identified, usually in our setting, by clinical testing. The category 2 are really the ones where we're trying to figure out how this might be useful for families. Category 3 is essentially a list of mutations, other cancer genes that are not currently thought to be as targetable. It's essentially a consensus list, such as the Sanger Center consensus list with some pediatric cancer genes added. And then category 4 is everything else. So mutations that if you go into PubMed or you start to read about that specific gene and mutation, you have no idea what that means in terms of cancer based upon our current biologic understanding. So the idea is essentially the reports give some prioritization, also gives some explanation of what's known about the biology for each mutation and what trials might potentially be available. So from the tumor exome results, we identify the expected genes that we figure to see from pediatric solid tumors. And enough research sequencing has now been done in numerous tumor types that these are not a surprise. So beta-catenin, P53, BRAF, et cetera. What I would point out is how low the frequency is for these various mutations. So this doesn't include copy number data. It doesn't include other types of data about translocations, et cetera, that might increase the yield of potentially relevant mutations. But the fact of the matter is even the most common mutations that are identified by exome sequencing in these pediatric cancer patients are only present in a handful of % of patients. And the fact of the matter is even more are present in mutations in just a single patient out of 100, for example. So, for example, we've seen singletons with potentially clinically relevant ALK mutations, RAS mutations, JAK2 or 3 mutations, all ones that on paper one can make the case might be of theoretical interest in terms of dictating therapies. But this is the reality. It's not a couple very frequently mutated genes. It's more this more complex spectrum of mutations. This shows that in a slightly different way. It's a bit difficult to read, but essentially on the y-axis is the number of mutations identified in an exome, and then each of the various columns is a different patient for the first set of about 67 cases that we looked at. And it's color-coded, each of the bars, based upon the same schema we were just looking at, where the red is the most clinically relevant, the gray is category 4 or non-cancer genes. And what you can see, first of all, is that there are not that many mutations detectable by an exome, so on the order of 5 to 10 for pediatric cancer. The second thing that's obvious is that the vast majority of this is gray, meaning the vast majority of mutations that we identify are currently thought to be in genes that we don't have any good understanding of how they relate to cancer. So in terms of future research study, in terms of future analyses, identifying which of those genes and which of those mutations might actually be relevant to cancer, how they're relevant, and what the clinical implications might be is obviously a hugely important area of research. You can see that there's some small number of, you see the yellows, the oranges, a pretty rare red, where there is for each patient some potentially clinically interesting result. And those are the ones that we're trying to look at more. This shows that same data in a slightly different way from an individual patient perspective, the same color schema. So if you took a patient, half of patients have no mutations whatsoever that are in what we understand to be a cancer-related gene. So for half of these patients, the test is completely useless in terms of identifying cancer gene mutations. There are a small number, as I said, that have what we call the category 1 or clinically relevant. There's about a quarter of them who have mutations that we think are potentially clinically relevant. There's another quarter that are in cancer genes, for example, some of the tumor suppressor genes that we really don't understand how to target at the moment. So this gives an idea for any of these individual tests what the yield might be. And the yield will be more if we're including genome data. The yield will be increased by adding copy number and amplifications and deletions, which we're doing. But the fact of the matter is, it's a challenge in terms of interpreting what these results mean for a patient. So I'll just show a couple of examples of some of the data. So this was a 14-year-old girl with a metastatic osteosarcoma. It's a bone tumor. This is a cross-section of bone. This whole white area here is the tumor. You can also see there's a little dot of white here, which is a lung metastasis. This is the histology of osteosarcoma. And this is, I like to show, is an example of some of these tumors and the complex genomic architecture and genetic architecture there is, and that you can see there's a huge number of variant chromosomes present in this patient. So we did whole exome sequencing. This is the data down below. So on the bottom, the tumor read, some selection of tumor reads. On the top, some selection of normal reads, showing essentially virtually all of the tumor reads have a frameshift mutation in the TSC2 gene, or tuberous sclerosis complex 2 gene. That's important, obviously, because it's related to the mTOR pathway, and in fact, patients who have the genetic disease tuberous sclerosis develop brain tumors, and there's actually an FDA-approved drug, an mTOR inhibitor for use in those patients that's been proven to be of benefit. So the question here is, for this patient with a poor prognosis and this mutation that doesn't have the genetic syndrome but has a tumor essentially has that same genetic alteration, could we potentially use one of these inhibitors for that patient? This is an example of a 7-year-old boy, a malignant liver tumor. So this is a cross-section of the abdomen. This whole thing here is essentially the liver tumor. This is a CT scan showing this patient also has a lung metastasis, so essentially an incurable patient. This is a case where the histology was not that revealing. All this pathologist could say was it's a malignant liver tumor. They couldn't tell us what type. In this case, a mutation was found in the NRAS gene, which is known to be pretty frequent in hepatocellular carcinoma, but less frequent and not so well known in hepatoblastoma, or the pediatric liver tumors. And so this is obviously a case where targeting RAS has been a huge challenge for oncology in terms of which of the specific pathways should be targeted downstream, what combinations should be used. And in theory, there's things that can be done, and this child was actually enrolled on a relevant trial. This is a 14-year-old boy who had a brain tumor called Medioblastoma. This is a cross-section of the head, brain tumor back here in the posterior fossa and cerebellum. This is what a small round blue cell tumor. And this is a case where the findings had both diagnostic and potentially prognostic and therapeutic uses. So there's a subset of medioblastoma, which are Wnt pathway tumors. So the finding of a beta-catenin mutation, a beta-catenin mutation plus a DDX3X mutation puts them in a specific subgroup with specific known prognostic implications. There were p53 mutations found on both alleles. And then there were also mutations in ARID1B, so a chromatin regulator, as well as FOXO3, which is related to the PI3 kinase pathway. So these are just examples showing we can find information that's of diagnostic, prognostic, and in theory, therapeutic use. But the challenge is when to use it for the patients. Do we use it at the time of diagnosis? Do we add it to their current therapy? If they relapse, do we try and enroll them on trials of these agents? That's where the practical limitations that you heard about in the previous talk come into play. The second thing I'd say is that we're under no illusions that exome sequencing is going to be the test of choice for these patients. So it's an example of the type of genomic studies that we can do and the types of challenges we face. So we do also ask permission from the patients outside of the clinical arena to do RNA sequencing, genome sequencing, copy number analyses, et cetera. In addition, we get blood samples, as I alluded to, from the parents so that we can do some of these studies, both to test what the actual genomic tests that we might want to use might be, but then also to see how we can start to study tumor heterogeneity, resistance and response, et cetera. So we've started to try and incorporate RNA sequencing, for example, into these data, both for identification of translocations and fusion genes, which we've done in a significant number of patients, but then also, for example, try to better interpret the exome data. And this is one of our real needs in terms of bioinformatics and integrating types of genomic data. So this shows, for example, a number of variants that were identified that we can call category 4 variants, meaning the ones where we don't really know what they mean for cancer. And the blue bars are for each variant, the tumor variant ratio that was seen in the genomic DNA. The red is what was seen when we did the RNA sequencing and looked at the cDNA. And so you can see, although there's some of the mutations that are clearly expressed and seen to a similar or significant fraction of cells at the RNA level, there's a number of these that were present in the tumor but are just not even expressed, giving us even less probability probably that they're important mutations for the biology of the tumor. We're also spending a lot of effort in parallel to develop preclinical models, both cell lines and mouse xenografts. So for example, all the brain tumors that we collect, they're also injected into these mice and develop these orthotopic xenograft models that are molecularly characterized. So we know for that patient what the mutations might be, and then we have a model for that patient. And we can go ahead and start to test hypotheses about whether specific targeted agents might actually work. Currently takes too long for clinical purposes. So on the germline side, there's obviously a number of findings that we can see as well. So I'm an oncologist. We've always thought about the tumor and what this might mean for treatment, but the fact of the matter, there's a whole bunch of other data here. So this kind of schematically shows that for exomes. So we have a tumor report where essentially we have enough problem or enough challenge identifying what the clinically and biologically relevant mutations are. On the germline side, we can both have pathogenic cancer susceptibility mutations, variants of uncertain significance in cancer genes, other pathogenic mutations that are not related to cancer. For example, this is a sodium channel mutation that causes long QT syndrome. We can get pharmacogenomic variants. We can also see information about recessive carrier gene status. And this is all the things that are in our cases. So as an example of a 14-year-old girl with a glioblastoma, when we had first talked to the family, they said, no, we had no family history of cancer. When our genetic counselors talked to the family, they actually saw that there's a pretty significant family history of cancer in mom's side of the family in the next generation. And this is a case where doing the exome sequencing found an MSH2 or a mismatch repair gene defect that explains this family history of cancer. Actually has practical implications in terms of surveillance testing and prevention strategies, and it's something that's already been used to test other family members and do various screening. So we see a number of these types of data results, some of which were clinically suspected, some of which were not, in terms of pathogenic cancer mutations. We also see a number of recessive cancer mutations where we see one alteration, but then we don't know what the other one is, and we don't know whether that's actually a family that is affected by that or not, and so further workup, both in terms of the function as well as other genetic testing, is underway. Finally, some of ththese patients also have other medical problems beyond cancer. So for example, we have a patient who had liver disease, end-stage liver disease, cirrhosis, and then developed a hepatocellular cancer, and there'd been a huge diagnostic journey to try and figure out what the explanation was for that liver disease. And so it turned out to be a mutation in a gene called TJP2. Similarly, a patient with renal disease had their disease explained, and others as well. Then the other part is what was also alluded to in the last talk, was there's incidental findings. So there's data that's unrelated to the patient's cancer or anything that we know about how their clinical situation. So these are some of the examples of those. So there's mutations, and I'm talking about what would be considered pathogenic mutations, not variants of uncertain significance. Mutations that cause long QT syndrome, which can cause cardiac sudden death, DSP and a number of cardiomyopathy genes, mutations causing familial hypercholesterolemia, and things that there's actually other interventions that should be taking place. Looking at a larger data set formed through our lab shows that about 5% of cases, 5% of exomes done will have one of these types of incidental findings. And so that's obviously something that needs to be taken into account. In addition, there's just huge other amounts of unexplored data within the exomes. So, for example, the typical patient has 3 variants of uncertain significance in cancer genes. So these will be missense mutations in NF1, these will be missense mutations in other cancer genes. All of our patients have them. We're doing studies to try and determine whether we see them more often than we'd expect. But the fact of the matter is developing algorithms and improved methods to predict which of these are important is an important area of further follow-up. We also see the recessive carrier mutations that we expect. Just as one aside, we do actually give our families, although all the other types of data I've talked about are part of the reports, and we tell families when they're enrolling that if you don't want these types of data, you should not enroll in this study. This is the one case where we give them a choice. We say, are you interested in having recessive carrier status as part of these results or not? The fact of the matter is the vast majority of our families and patients do want that information. And so we see what we'd expect there. We also see some pharmacogenetic variants. So if you bin all this together from the exome sequencing, you can make a case that you find potentially clinically relevant findings in a significant minority of patients. It's still not a huge number, but between 30 and 40% will have some germline or tumor finding that's of potential clinical actionability. So to wrap this up, I'd say that one of our goals has been achieved, which is developing a clinical pipeline for the process of doing genomics. We can find mutations that are potentially relevant in a significant number of patients, but we're trying to figure out how to find more. The diversity of mutations here says that the exome test is probably not going to be sufficient. We need to figure out other ways to incorporate other types of genomic data, which is one of the challenges. So for example, bringing exome data and RNA sequencing data together, and looking further at heterogeneity will be important. As I mentioned, there's huge family and patient interest in these types of approaches, which puts a significant onus on us to do it in a responsible way. We do have a mechanism set up that we have genetic counselors and facilities and follow-up available to follow up on those types of results. Obviously, there are huge interpretive challenges that remain here, both in terms of what we consider the pathogenic or clinically important genes, but then also in terms of this vast array of data that we really don't know how to interpret at this point. And we're trying to spend more time figuring out not only how to use those, but what the preferences are for these patients. Currently, we're using the same test to design prospective clinical trials to try and see how often we can find anything that'll actually help our kids. So this is a partial list of the folks involved in the study. It's sponsored by the NHGRI and NCI under their Clinical Sequencing Exploratory Research Project. Between the clinicians, the sequencers, the ethicists, the pathologists, and surgeons and neurosurgeons, it's a huge number of people. So I'll stop there, and I appreciate your attention. Thank you.